So this is the second video from lecture 2, and it's about bacterial growth. I want to do this in one video, but I don't know if that's going to happen or not. So let's find out together. Let's go explore the future. It's happening now. Okay, so bacterial growth is what we say when we are talking about a bacterial population, not an individual, usually. So the term bacterial growth refers to the increase in the number of cells in a population. So we do think of bacteria as little individual cells, but we almost always see them in huge groups. So we care more about how many of them there are than we do about what any one individual cell is doing. So bacterial growth happens when lots of cells divide. Bacterial cells divide through binary fission, so they split into two daughter cells, and this is what I was alluding to in the first video when I was talking about the cell shapes and cell morphologies and how they stay connected to each other. This is a lot like the cell cycle in humans. Bacteria have to do the same thing human cells do, with the exception that bacteria don't have nuclei or organelles, so they don't have to do mitosis and separate the chromosomes and make a new nucleus. I mean, that takes a lot of work for one of our cells, and bacteria never have to do that. But they have to do the other parts of the cell cycle, or their own types of cell cycle. They have to do what's similar to G1, where they recover from division and decide whether or not to divide again. Spoiler, they always try to divide again. Then they have to do what we would do in S phase, where they would make a new copy of their DNA, and they'd make all the proteins they need to do that. Bacteria tend to grow, they tend to reproduce faster than our cells do. And so sometimes they have to kind of mix these things and do them all at the same time, but they have to accomplish the same goals. And ultimately they have to get big. The cells have to get big enough that when they split in half, they're still complete cells. And so that process of getting bigger we call elongation. So elongation is the increase in size that they do before they divide. But growth is the bulk. It's a change in the bulk number of cells in a population. It's not the size of any one cell. So the way elongation works tends to be at some location on the cell. There is what we call a growth ring. And at that growth ring, they will make new cell wall and cell membrane. And so the cell will grow away from that ring in both directions. There are other ways they do it, but that's the one we understand the best. And that's elongation. I won't get into it in that much detail, but just as we learn more things about cells, even in today's lecture, we're going to learn about cell walls and cell membranes. Just understand they have to build these very quickly as they elongate. And they have to navigate what happens when two cells split. They have to do that without breaking open. So, since they divide, every time they do that, they double their numbers. So one thing we measure in microbiology is the doubling time. We measure how long it takes a population of bacteria to double. And this is a widely varying number in different situations. It's really variable. It depends a lot on the environment the cells are growing in. If they run out of nutrients, they can't divide and they slow down a lot. If they encounter something toxic, they have to keep fixing the proteins that get broken down and they slow down a lot. And some cells seem to specialize in dividing quickly, and these will try to take over their environment and poison all the other cells. And others specialize in growing in really terrible environments that most cells never could grow in. And the best they can do is divide very, very slowly. So the fastest I know of is 7 minutes. And the bacterium that does that can do that in beef gravy, hot beef gravy, in the absence of oxygen. It does that. The slowest I know of takes about a month, roughly, to double. And there's a lot of interesting stuff going on there that we won't get into. But in terms of the numbers we need to think about to do this math stuff, what I want you to remember is that the doubling time is the same when we're going from 2 cells to 4 as it is when we're going from 3 trillion to 6 trillion. So as long as the cells have all the nutrients they need and they're not being poisoned by their waste products, it really is as fast for 2 cells to divide as it is for 3 trillion cells to divide. And so that doubling time will be constant across a wide range of population sizes and numbers. So here is what we see when we graph the population of bacteria over time. And we're going to look at some graphs here. And what I want is for you to be able to read these graphs and understand what you're looking at. And there are 3 different types of graph I'm going to show you. In every case, we are looking at the numbers of bacteria in a population that is growing. And so it's growing, as we say, exponentially, where it has a constant doubling time. So let me walk you through kind of what we're seeing in this graph and ignore this question for now. Let's just look at this graph. So this axis is the population in cells per milliliter. So if this were growth in a test tube, we would be measuring how many cells are there in a milliliter of the liquid. So you could think of it as analogous to a concentration of a chemical, but instead of moles per liter, it's cells per milliliter. And this axis would be time, and that would depend on how fast or slow the cells are at dividing. So I made these graphs in Excel, and it does some weird things with scientific notation. So I want to show you what this is, if you haven't worked with Excel a lot before. It doesn't want to use subscripts or superscripts in these labels. You can't actually make it do that in any of these kind of labels. So they came up with a different way of doing it, where if this is 1 times 10 to the 6th, that's what this is. When they say 10 to the 6th power, they just write e plus 06, and that means 10 to the 6th power. This is 10 to the 5th power, 10 to the 5th power, 10 to the 5th power. So we go from 0 up to 2 times 10 to the 5th, up to 4 times 10 to the 5th, 6 times 10 to the 5th, 8 times 10 to the 5th, 1 times 10 to the 6th. So we've gone like 2, 4, 6, 8, 10. 10 times 10 to the 5th power is 1 times 10 to the 6th power. So it's like 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. When we get to 10, the power of 10 goes up. And then this is 1 times 10 to the 6th, 1.0, 1.2 times 10 to the 6th. So each time we go up, we are increasing by the same amount. This is a linear scale. Each time we're going up by this amount, 2 times 10 to the 5th. So if we go from 2 times 10 to the 5th up and we add 2 times 10 to the 5th, we get 4. If we add 2 times 10 to the 5th to 4, we get 6. So we really are going up by the same amount each time. And if I put this in English, that's 200,000, 400,000, 600,000, 800,000, 1 million, 1.2 million, or 1,200,000. Okay, that's a lot of that. So what do we see in terms of the bacterial population over time? Well, it goes up in this exponential way. And if you're curious, these numbers are idealized numbers. If I remember correctly, the numbers I put in here double every 20 minutes. So if this is an hour, that's 60 minutes long. So 20 minutes is 1/3 of that. So this is 20 minutes, 20 minutes, 20 minutes. Okay, let's find out if I am correct about that. Let's see if this number is double this number. And then we'll see if this number is double this number. So what is this number? It is a little over 1 times 10 to the 6th. And it's much less than 1.2 times 10 to the 6th. So this is 1 million. It's closer to 1 million than it is to 1,200,000. So let's say 1.05 times 10 to the 6th power, 1.05, something like that. What's this? This is between 4 and 6 times 10 to the 5th. So this is around 5 times 10 to the 5th. So if you multiply 5 by 2, you get something like a little over 10. So this is double this. This is a little over 5 times 10 to the 5th. What's this? It's a little over 2. It's between 2 times 10 to the 5th and 3, so like 2.5 times 10 to the 5th. Double that, you get 5 times 10 to the 5th. Something like that. So I just wanted to convince you and myself that it is doubling at a constant rate of 1 doubling every 20 minutes. So if I asked you to do that same analysis for this point and this point, I don't think you really could. You really can't, on this graph, read what this is. And that's something we run into when we're graphing bacterial growth. That's kind of the purpose of this exercise, is to show you that. When we have exponential growth, we could think of it going back in time as dividing by 2, divide by 2, divide by 2, divide by 2, divide by 2, and eventually we get to a small enough number that on this scale it's just essentially 0. But it isn't 0. It isn't 0 at all. And I'm going to show you on the next graph what these numbers actually are. So on the next slide, on the next slide, I've graphed the same data on a logarithmic scale. So this is like logarithmic graphing paper. And what we find is a couple weird things. First, now this is a straight line, whereas back on this previous slide, it was this curve. Now, if I look at these first few data points, I can explicitly figure out where they are on this scale. Whereas if we go back, these all looked like 0 on this scale. So this logarithmic graphing paper, it works a little bit differently. It's laid out in a different way. So the x-axis is the same. It's just time, 1 hour, 2 hours, 3 hours, 4 hours. But this axis is very different. So we are going from this line to this line. We go from 10 to the 3rd power to 10 to the 4th power. When we go from here to here, we're going from 10 to the 4th power to 10 to the 5th power. And from here to here, 10 to the 6th. So in English, that's from 1,000 to 10,000, from 10,000 to 100,000, from 100,000 to 1 million. So each big line you go up is a factor of 10. And they filled in some lines here to help you figure out what is between these powers of 10. And these look kind of weird. Let me show you how to read these on the next slide. What I've done on this slide is only graph a small part of the data. So we're only going up to 2 hours. And if we go back to the previous slide, we see if we get to 2 hours, that's only to here. And the numbers only get up to around 10 to the 5th power in that time. So we're really only graphing this much. So that's what it looks like. And what I want to show you is how to read this. So again, these big lines are powers of 10. This is 10 to the 3rd, 10 to the 4th, 10 to the 5th power. And those are like 1,000, 10,000, 100,000. So how do we figure out what is between 1,000 and 10,000? Well, that's what these little grid lines are for, and they have this weird pattern. And the way to read them is a little bit different than what you'd be used to in linear or regular graph paper. My brain just disappeared for a second. So here's how we read this. We start with 1,000, or 1 times 10 to the 3rd power, 2,000, 3,000, 4,000. So 2,000, 3,000, 4,000 is the same as 2 times 10 to the 3rd, 3 times 10 to the 3rd, 4 times 10 to the 3rd, 5,000, 6,000, 7,000, 8,000, 9,000, or 9 times 10 to the 3rd, and then 10,000, or 10 times 10 to the 3rd power. But once we get here, the lines stop, and we wait until we get another big line, and that's 2 times 10 to the 4th, and then 3 times 10 to the 4th, and 4 times 10 to the 4th. So that's like 20,000, 30,000, 40,000. So these numbers, these lines in English go from 2,000, 3,000, 4,000, 5,000, 6,000, 7,000, 8,000, 9,000, 10,000, 20,000, 30,000, 40,000, 50,000. So it does take a little bit of practice to read these graphs. But if you put some time into it, you will be able to do it. And the beauty of this is that we can graph bacterial growth on one of these graphs, and we can read every point. We don't have to guess at what the numbers are because we can plot them all this way. And also, it comes out in a straight line, and there's some math we can do with the slope of this line that we're not going to do in this class, but it is possible to figure out the doubling time just using the slope of this curve on this logarithmic paper, or logarithmic graph. Okay, so we go on to 14, and this is a challenge for you. Which of these points is closest to 6 times 10 to the 4th power? Go ahead and pause this, and I will solve all the math questions on the last slide. I'll make a separate video for that. I don't want you to watch that yet. I want you to do this on your own to the extent possible. And what I'm going to tell you is you can do it. Whoever you are able to do this, but you have to put some time into it. So do that now, and then we'll move on to the next slide once you've done that. I changed my mind. I'm going to end this video here because this video got kind of long. So I will see you at the next video.